This is a presentation on the mechanisms of antibiotic resistance observed in vancomycin-resistant Enterococci. During this presentation, we will discuss Enterococci and the typical infections that this microorganism causes, the emergence of vancomycin-resistant Enterococci and the dominant isolates encountered in the clinical setting, the introduction of vancomycin into use and the actions this antibiotic has on bacterial cells. The mechanisms of antibiotic resistance will also be examined. And finally, we will discuss how vancomycin resistance develops and how it is spread amongst bacteria. Now, Enterococci are gram-positive bacteria normally found in the intestinal tract of humans and animals. Because Enterococci is part of the normal human flora, it most often does not adversely affect healthy individuals. However, in patients with suppressed immunity, this organism can cause significant problems. As a result, serious enterococcal infections are most often observed in intensive care units, transplant recipients, or oncology patients. Sites of enterococcal infection often include the urinary tract or other catheterized sites, the heart and bloodstream, the pelvic or abdominal cavity, and wound sites. Vancomycin-resistant Enterococci, or VRE, was first reported in England in 1988. This case was soon followed by reports of additional cases of vancomycin resistance in Europe and the United States. The first Australian case of VRE was encountered in a Melbourne hospital in 1994 and has since been found in all Australian states. While the Enterococcus genus includes more than 17 species, many of these are not pathogenic to humans, and vancomycin resistance is normally encountered in two species, Enterococcus faecalis and Enterococcus faecium. Between these two species, Enterococcus faecalis accounts for approximately 80 to 90% of VRE isolates, while Enterococcus faecium accounts for between 5 and 15%. The genetic basis for vancomycin resistance is caused by several phenotypes known as VAN A, VAN B, VAN C, VAN D, VAN E, and VAN G. While these phenotypes provide varying levels of vancomycin resistance, they all work on similar resistance mechanisms. Now, these graphs provide an indication of the increasing prevalence of VRE. The figure on the left shows the increase in prevalence of vancomycin-resistant isolates of Enterococcus faecium in Australia between 1995 and 2005. Over this 10-year period, VRE prevalence has risen from virtually zero to between 7 and 9%. The figure on the right shows similar data for the United Kingdom between 1994 and 1997. Note the prevalence of VRE in the United Kingdom is significantly higher than in Australia for a similar time frame, but it does appear stable in later years. Vancomycin first received approval from the US Food and Drug Administration in 1958 after being isolated from the soil bacterium Streptococcus orientalis. This drug was introduced in response to increasing prevalence of penicillin resistance encountered in staphylococcal infections. Vancomycin is a glycopeptide class antibiotic. This group of antibiotics are most effective against gram-positive organisms, specifically gram-positive cocci. As mentioned earlier, the first reported case of VRE was in 1988, some 30 years after the drug's initial introduction. This is in line with the steady increase in vancomycin use that occurred over this time period. This diagram shows the different actions of antibiotics on bacterial cells. These include inhibition of cell wall synthesis, inhibition of protein synthesis, inhibition of nucleic acid replication, injury to the plasma membrane, and inhibition of essential metabolite synthesis. The main actions of vancomycin is the inhibition of cell wall synthesis, but vancomycin also alters protein synthesis and membrane permeability. We will now examine how vancomycin inhibits cell wall synthesis. Here we see a diagram of the bacterial cell wall, which is made up of peptidoglycan monomers. Between these monomers are peptide chains that form an essential part of the cell structure. During cell wall synthesis, a cross-linking enzyme attaches to the two peptide chains and forms a peptide cross-link between them. These peptide crosslinks are what gives the bacterial cell wall its strength, and this is exploited through the actions of vancomycin. When vancomycin enters the cell, it binds to the residues located at the end of the peptide chains to prevent the cross-linking enzyme binding and forming peptide crosslinks. This causes weak points to form in the cell wall where peptidoglycan is lacking. The cell membrane then bulges out as water enters the cell and eventually lyses, causing bacterial cell death. In vancomycin-resistant bacteria, the residues on the end of the peptide chain to which vancomycin binds are substituted with a different residue. This prevents vancomycin binding with the peptide chain, but still allows the cross-linking enzyme to bind to the peptide chains and create the peptide crosslink. So what actually causes antibiotic resistance? Well, the changes discussed in the last slide are the result of changes in bacterial DNA and the products they code for. This normally occurs through a random mutation of the DNA in bacteria and is then spread to other bacteria in a number of ways. One way in which antibiotic resistance is spread by selective pressure through antibiotic use. This is often the result of inappropriate or excessive use of antibiotics, but this only accelerates the development of resistance, as resistance is bound to develop in time anyway. Another method of spreading resistance is through the sharing of resistance genes between bacteria. These two means will now be examined. We'll first discuss selective pressure through antibiotic use. Here we see a representation of a population of bacteria. The blue bacteria are susceptible to vancomycin, where the orange bacterium is resistant. When vancomycin is introduced, all of the blue bacteria are killed, but the orange bacterium is unaffected due to its resistance. With the other bacteria gone, the orange bacterium has no competition for food or space and is able to freely reproduce additional bacteria who also possess resistance to vancomycin. When vancomycin is introduced at a later time, it has no effect on the population because all the bacteria are now resistant. Next, we will discuss conjugation. The genes responsible for bacterial resistance are commonly found on bacterial plasmids. Plasmids are fragments of genetic material separate from the bacterial chromosome. Now, bacterial cells with antibiotics resistance are able to share this resistance with other bacteria through a process called conjugation. During conjugation, two proximal bacteria are joined by a conjugative pilus, which facilitates the passage of genetic information between them. The bacterium with the resistance replicates its plasmid and then passes the copy onto the other bacterium. This results in both bacteria now being resistant. In summary, Enterococci are part of the normal human intestinal flora and cannot ever be completely eliminated because of this. Antibiotic resistance often develops in Enterococci native to an individual, but can also be acquired during hospital admission. Antibiotic resistance is something that cannot be avoided due to the rapid rate at which bacteria reproduce and the effect this has on DNA mutation. Inappropriate use of antibiotics only acts to accelerate the development of resistance and shortens the shelf life of antibiotics. The most practical means of helping to prevent bacterial resistance is through effective infection control measures such as hand hygiene and environmental cleaning to prevent the spread of infectious organisms. This concludes the presentation. I thank you for your attention.